हेलो फ्रेंड्स, वेलकम टू माय यूट्यूब चैनल BEU हेल्पर। आज मैं आपके लिए लेकर आया हूं बायोलॉजी फॉर इंजीनियर्स के PYQ ईयर 2020 का कंप्लीट सॉल्यूशन फॉर थर्ड सेम ऑफ मैकेनिकल और सिविल ब्रांच। इस क्वेश्चन का कोड है 100301। इस क्वेश्चन के लिए दिए जाते हैं 3 आवर्स का टाइम। टोटल मार्क्स है 70 मार्क्स। टोटल 9 क्वेश्चन दिए गए हैं जिसमें सॉल्व करने हैं ओनली 5 क्वेश्चंस। 1 नंबर क्वेश्चन कंपलसरी है जो सारे ऑब्जेक्टिव है। इसमें 10 ऑब्जेक्टिव्स दिए गए हैं जिसमें सॉल्व करने हैं ओनली 7 ऑब्जेक्टिव्स। तो वीडियो को स्टार्ट करने से पहले मेरे इस वीडियो को लाइक एंड शेयर जरूर करें और मेरे चैनल को सब्सक्राइब जरूर करें जिससे मेरे सारे वीडियो आप तक आसानी से पहुंच सके। फर्स्ट ऑब्जेक्टिव है द टर्म बायोलॉजी वाज इंट्रोड्यूस्ड बाय। देन आंसर इज लेमार्क एंड ट्रेविरेनस। नेक्स्ट बी नंबर द टाइम डिपेंडेंट परमानेंट डिफॉर्मेशन इज कॉल्ड क्रीप। Next, which of the following is not present in animal cell? Then answer is cell wall. Next, normal human blood pressure is 120/80. Next, which ancient Indian physician is known as the father of surgery? Then answer is Sushruta. Next, the extension of gene was first suggested by. Then answer is Gregor Mendel. Next, which of the following animals has the longest lifespan? Then answer is tortoise. Which of the following is the reason for orange color of carrot? Then answer is it contains carotene. Next, Rh factor of the blood was discovered by the scientist Landsteiner and Wiener. The suitable temperature to transport viral culture is 5 degrees Celsius. नेक्स्ट सेकेंड नंबर में सभी का आंसर ब्रीफ में देना है। फर्स्ट है व्हाट इज द वर्किंग प्रिंसिपल ऑफ ह्यूमन आई एंड डिजिटल कैमरा। ह्यूमन आई एक नेचुरल ऑप्टिकल इंस्ट्रूमेंट है जो ह्यूमन बीइंग को देखने के योग्य बनाता है। ह्यूमन आई रिफ्रैक्शन ऑफ लाइट के प्रिंसिपल पे वर्क करता है और ये जस्ट कैमरा के प्रिंसिपल पे ही वर्क करता है। जैसे कैमरा में फिल्म होते हैं वैसे ह्यूमन आई में रेटिना होता है। जैसे कैमरा में डायफ्राम होता है वैसे ह्यूमन आई में आइरिस होता है। जैसे कैमरा में एपर्चर होता है वैसे प्यूपिल होता है। जैसे कैमरे में लेंस होते हैं वैसे ह्यूमन आई में भी लेंस होते हैं। जैसे कैमरा में बैक पेंट होता है वैसे कोराइड होते हैं। तो इसे आप अपने अकॉर्डिंग और भी डिफाइन करके लिख सकते हैं। नेक्स्ट, डिफाइन मेंडल्स लॉ। मेंडल्स लॉ also known as Mendelian inheritance are fundamental principles in the field of genetics that are formulated by Gregor Mendel, often referred to as the father of genetics. Mendel conducted groundbreaking experiments with pea plants in the 19th century and established the fundamental law of heredity. तो टोटल 3 लॉ दिए गए हैं। आप इसे डिफाइन करके भी लिख सकते हैं। लॉ ऑफ सेग्रीगेशन, लॉ ऑफ इंडिपेंडेंट असॉर्टमेंट, लॉ ऑफ डोमिनेंस। These law provide a framework for understanding how traits are inherited from parents to offspring and how genetic variation are passed down through generations. They form the basis of classical genetics and have been validated and expanded upon by subsequent scientific research. Next, classify about the type of sugar. तो शुगर को 3 टाइप्स में बांटा गया है. फर्स्ट ऑन द बेसिस ऑफ टेस्ट्स, सेकेंड ऑन द बेसिस ऑफ रिड्यूसिंग पावर, थर्ड ऑन द बेसिस ऑफ स्ट्रक्चर. फर्स्ट है ऑन द बेसिस ऑफ टेस्ट्स. तो इसमें 2 पार्ट्स में बांटा गया है. पहला शुगर, जिसका एग्जाम्पल है ग्लूकोज, सुक्रोज, फ्रुक्टोज, ग्लाइकोज, इटीसी. देन सेकेंड नॉन शुगर, एग्जाम्पल स्टार्च, ग्लाइकोजन, सेलुलोज. नेक्स्ट ऑन द बेसिस ऑफ रिड्यूसिंग पावर, ये भी 2 टाइप्स में डिवाइड किया गया है. फर्स्ट है रिड्यूसिंग शुगर, जिसका एग्जाम्पल है अल्डोज टाइप शुगर. देन सेकेंड नॉन रिड्यूसिंग शुगर, जिसका एग्जाम्पल है केहोज टाइप शुगर. नेक्स्ट ऑन द बेसिस ऑफ स्ट्रक्चर, इसके 3 टाइप्स हैं. पहला मोनोसेक्राइड्स, दूसरा ओलिगोसेक्राइड्स, थर्ड पॉलीसेक्राइड्स. इसे आप अपने अकॉर्डिंग एक्सप्लेन भी कर सकते हैं. नेक्स्ट, हाउ कैन यू से दैट DNA is a genetic material? The molecule that bears genes is passed from parents to children and provides instruction for the growth and functioning of living organisms. They determine your eye color, facial features, and other physical attributes. The quantity of DNA is enabled in diploid cell and half in gamete. DNA has self-replicating power. The replication of DNA occurs in S phase of interphase. Next, third number, write the answer in very brief of the following. First, list the properties of genetic code. Then answer is, the genetic code refers to the set of rules that govern the translation of the DNA or RNA sequence into the corresponding sequence of amino acids during protein synthesis. Properties of genetic code are: first, triplet code; second, non-ambiguous; third, universal; fourth, degenerate code. Next, non-overlapping code. Next, comma-less. Next, start and stop codons. Next, polarity. Next, B number, define the term of primary structure of protein. The primary structure of a protein refers to the specific sequence of amino acids that make up the protein chain. It is a linear arrangement of amino acids which are linked together by peptide bonds to form a polypeptide chain. The primary structure is determined by the sequence of nucleotides in the gene encoding the protein, which is transcribed into messenger RNA, which is also known as mRNA, and then translated into the corresponding sequence of amino acid. Next, C. List the law of thermodynamics. The flow of energy in any ecosystem is based on 2 important laws of thermodynamics, which are as follows. At first law of thermodynamics, the amount of energy in the universe is constant. It may change from one form to another, but it can neither be created nor destroyed. Example, light energy can neither be created nor destroyed as if passed through the atmosphere. At second law of thermodynamics, non-random energy, if mechanical, chemical, and radiant energy cannot be changed without some degradation into heat energy. The change of energy from one form to another form takes place in such a way that a part of energy assumed waste form. Next, write the example of single cell organism and multicellular organism. Single cell organism. Single cell organisms are also known as unicellular organisms or living organisms that consist of a single cell as their entire body. They are considered the simplest and most basic forms of life. Example, paramecium, amoeba, euglena, bacteria, yeast, etc. And next, multicellular organism. Multicellular organisms are living beings composed of multi-cells that work together to form tissue, organs, and organ systems. They exhibit a higher level of complexity compared to single-cell organisms. Example, all plants, rat, monkey, human, insect, worm, etc. Next, for number, write the answer in sort of the following. At first, differentiate between prokaryote and eukaryote organisms. Prokaryotic cell is always unicellular, but eukaryotic cell are unicellular और multicellular. प्रोकार्योटिक सेल के डायमीटर का रेंज होता है 0.2 टू 2.0 माइक्रोमीटर्स, लेकिन यूकार्योटिक सेल के डायमीटर का रेंज होता है 10 टू 100 माइक्रोमीटर्स। प्रोकार्योटिक सेल में सेल वॉल प्रेजेंट रहता है और वेरी कॉम्प्लेक्स होता है नेचर में। यूकार्योटिक सेल में बहुत कम सेल वॉल पाए जाते हैं, अगर सेल वॉल पाए भी जाते हैं तो वो बहुत सिंपल होते हैं, केमिकल नेचर के। प्रोकार्योटिक सेल में न्यूक्लियस अब्सेंट होता है एंड इंस्टेड न्यूक्लियोटाइड इज प्रेजेंट, लेकिन यूकार्योटिक सेल में ट्रू न्यूक्लियस जो होते हैं प्रेजेंट होते हैं। प्रोकार्योटिक सेल में डीएनए अरेंज होता है सर्कुलर शेप में, लेकिन यूकार्योटिक सेल में डीएनए अरेंज होता है लीनियर शेप में। प्रोकार्योटिक सेल में साइटोप्लाज्म प्रेजेंट होता है, लेकिन सेल ऑर्गेनल्स की कमी होती है, लेकिन यूकार्योटिक सेल में सेल ऑर्गेनल्स एंड साइटोप्लाज्म दोनों प्रेजेंट होते हैं। प्रोकार्योटिक सेल में माइटोकॉन्ड्रिया एब्सेंट होते हैं, लेकिन यूकार्योटिक सेल में माइटोकॉन्ड्रिया प्रेजेंट होते हैं जो पावर हाउस ऑफ द सेल कहलाते हैं। प्रोकार्योटिक सेल में राइबोसोम्स प्रेजेंट होते हैं छोटे साइज में एंड स्फेरिकल इन शेप में, लेकिन यूकार्योटिक में राइबोसोम प्रेजेंट होते हैं बहुत लार्ज और लीनियर शेप में। प्रोकार्योटिक सेल में एंडोप्लाज्मिक रेटिकुलम और लाइसोसोम्स और सेंट्रोमर्स सभी एब्सेंट होते हैं, लेकिन यूकार्योटिक सेल में एंडोप्लाज्मिक रेटिकुलम, लाइसोसोम्स और सेंट्रोमर्स सभी प्रेजेंट होते हैं। प्रोकार्योटिक सेल में प्रोकार्योट्स में प्लाज्मिड्स पाए जाते हैं, लेकिन यूकार्योटिक सेल में यूकार्योट्स में प्लाज्मिड्स बहुत कम पाए जाते हैं। इन प्रोकार्योटिक सेल सेल डिवीजन occurs through binary fission, but in eukaryotic cell division occurs through mitosis. प्रोकार्योटिक सेल में फ्लैगला बहुत स्मॉल होते हैं, लेकिन यूकार्योटिक सेल में फ्लैगला बहुत लार्ज होते हैं साइज में। प्रोकार्योटिक सेल का अगर एग्जांपल देखें तो होगा बैक्टीरिया एंड आर्की। यूकार्योटिक सेल का एग्जांपल होगा प्लांट्स एंड एनिमल सेल्स। नेक्स्ट बी नंबर, व्हाट आर द रिसेसिव ट्रेट्स इन लिविंग ऑर्गेनिज्म? A recessive trait that is expressed when an organism has two recessive alleles or form of a gene. Every organism that organizes its DNA into chromosomes has two alleles for a trait, one from their mother and one from their father. Example, the allele for 5 eyes, dwarf plant and white flower. Individual with recessive allele for a cleft chin, dimples or freckles do not have these traits. Next 5 number, write the answer in sort of the following. At first, explain about the Brownian motion. Brownian motion refer to the random movement displayed by small particles that are suspended in fluids. It is commonly referred to as Brownian movement. Brownian motion is named after Scottish botanist Robert Brown, who first observed that pollen grains move in random direction when placed in water. The size of particles is universally proportional to the speed of the motion. Brownian movement causes the particles in fluid to be in constant motion. Next B number, write the part of light microscope used for observation of cells. A light microscope, also known as an optical microscope, is a widely used instrument for observing cells and other microscopic objects. It consists of several essential parts that work together to magnify and illuminate The sample being observed. Here are the main components of a light microscope used for cell observation. Eyepiece or ocular lens. This is a lens closest to the viewer eyes. It typically magnifies the image further, usually by 10x or 15x, and is usually equipped with a diopter adjustment to accommodate individual difference in vision. Objective lens. The objective lens is a series of lenses located near the specimen being observed. Next stage. The stage is a platform where the specimen is placed for observation. Next, condenser. The condenser is located beneath the stage and focus light onto the specimen. Next, diaphragm. The diaphragm is part of the condenser and controls the amount of light passing through the specimen. Next, coarse and fine focus knobs. These knobs are used to bring the specimen into sharp focus. Next, arm. The arm is the curved part of the microscope that connects the base to the head. It provides support and stability to the microscope. Next, illuminator. The illuminator is a light source that provides illumination for the sample. At last, base. The base is the bottom part of the microscope that provides the stability and support for the entire instrument. Next 6 number. Write the answer in sort of the following. At first A number. Discuss how biological discovery of 20th century led to major investigation in medical science. The 20th century was transformative period in biological discovery leading to significant advancements in medical science. Numerous breakthroughs in understanding the molecular, cellular and genetic basis of life revolutionized our knowledge of biology and paved the way for major investigation in medical research. Discovery of DNA structure. In 1953, James Watson and Francis Crick elucidated the structure of DNA, the molecule that carries genetic information. The discovery laid the foundation for understanding the mechanism of inheritance and led to subsequent research in genetic, genomics, and molecular biology. Development of antibiotics. The discovery of antibiotics such as penicillin by Alexander Fleming in 1928 and subsequent antibiotic development effort revolutionized the treatment of bacterial infections. Next, identification of blood types. In the early 20th century, Karl Landsteiner identified different blood types A, B, AB, and O and discovered the compatibility and incompatibility of blood transfusions. Next, advance in immunology. The 20th century saw significant progress in understanding the immune system and how it defends against the pathogens and recognizes foreign substances. Next, mapping the human genome. In 2000, the completion of the Human Genome Project, an international collaboration to sequence the entire human genome, marked a major milestone. Next, stem cell research. The isolation and characterization of stem cells, particularly human embryonic stem cells, in the late 20th century opened new avenues in regenerative medicine. Next, understanding the molecule basis of cancer. Extensive research in the 20th century unraveled the molecular mechanism underlying cancer development and progression. Next, discuss about the Mendel's law of segregation and independent assortment. Mendel's law of segregation and independent assortment are the fundamental principle in the field of genetics that explain the inheritance pattern of traits from one generation to the next. Law of segregation. The law of segregation states that for any given traits, an individual organism inherits two alleles, one from each parent, and these alleles segregate during gamete formation. As a result, each gamete carries only one allele for a particular trait. During fertilization, when the gametes combine, the offspring receive one allele from each parent, thereby inheriting two alleles for the traits. Mendel's experiment with pea plants demonstrate this law. For example, when he crossed plants with purple flower and plants with white flowers, the first generation of spring all had purple flowers. However, in the subsequent generation, the purple and white flower traits appeared in a predictable ratio, approximately 3:1. The observation supported the idea that the allele for flower color segregate during gamete formation. Next, law of independent assortment. The law of independent assortment states that the inheritance of one trait is independent of the inheritance of another trait when these traits are located on different chromosomes or are assorting independently. Mendel's experiment with pea plant also revealed the law of independent assortment. He conducted experiments with plants that differed in two traits, such as flower color and seed texture. Mendel found that the inheritance of flower color and seed texture occur independently of each other, meaning that the presence of one trait did not influence the presence of other traits. Next, 7 number. Write the answer in short of the following. At first, A number. How can you say that proteins are transporters and receptors of a living cell? Cell needs to communicate with their environment in order to obtain nutrients, grow, divide, and respond to signals related to adaptation in changing psychological condition or stress. A very basic question in biology is how cell, especially of those organisms living in rapidly changing habitats, sense their environment. Apparently, this question is of particular importance of all living microorganisms. The critical role of receptors, transporters, and channels transmembrane of all type of alleles in signaling environment changes in well established. A relative new corner in environment sensing are called transceptors membrane. Proteins that possess both solute transport and receptors like signaling activities. Now the transceptors concept is further enlarged to include micronutrient sensing via the iron and zinc high affinity transporters of Saccharomyces and Cerevisiae. Interesting, what seems to underline the transport and or sensing functioning of receptors, transporters and transports is ligand-induced conformational alteration recognized by downstream intercellular effectors. Next, B number, differentiate between photosynthesis and transpiration in plant. At first, photosynthesis is a plant process that converts atmospheric CO2 into more complex organic compounds, especially sugar, using energy from sunlight. Transpiration is water taken up from the soil and washed through the stomata in the leaves. Photosynthesis is energy transformation process, and transpiration is not energy transformation process. Photosynthesis helps in formation of food. Transpiration helps in removal of excess water from plant body. Photosynthesis takes place through chloroplast. Transpiration takes place through stomata. Energy is required for photosynthesis, but in transpiration, no energy is required. Next, 8 number. How can you prove that biology is an important scientific discipline similar to mathematics, physics, and chemistry? Answer in detail. Biology is an important scientific discipline that is on par with mathematics, physics, and chemistry in terms of its significance and impact. Study of life. Biology is the scientific study of life and living organisms. It explores the structure, function, growth, evolution, and distribution of living organisms, ranging from microscopic cells to complex ecosystems. Next, interdisciplinary nature. Biology is an inherently interdisciplinary field that intersects with various other scientific disciplines. It draws upon principles from chemistry, physics, mathematics, genetics, ecology, neuroscience, and more. Next, medical advances. Biology plays a central role in medical science and healthcare. The study of biology has led to numerous breakthroughs in understanding disease, developing diagnostics, and designing effective treatments. Next, agricultural and food production. Biology is instrumental in enhancing agricultural productivity and addressing food security challenges. Next, conservation and environmental science. Biology play a crucial role in understanding and addressing environmental issues and conserving biodiversity. It helps in assessing the impact of human activity on ecosystem, monitoring endangered species, studying climate change, and developing conservation strategies. Next, evolutionary understanding. Biology provides a framework for understanding the evolution of life on Earth. The theory of evolution by natural selection, developed by Charles Darwin, is a fundamental concept in biology. Next, biotechnology and bioengineering. Biology has paved the way for significant advancement in biotechnology and bioengineering. These fields utilize biological principles and techniques to develop novel products, therapies, and technologies. नेक्स्ट 9 नंबर में ड्रॉ द फ्लो चार्ट ऑफ कर्व साइकिल। तो ये कर्व साइकिल का फ्लो चार्ट है एंड ये कर्व साइकिल के फ्लो चार्ट का एक्सप्लेनेशन है। एंड थैंक यू इस वीडियो को लास्ट तक देखने के लिए। आई होप ये वीडियो आपके एग्जाम के लिए बहुत हेल्पफुल होने वाली है, क्योंकि बीईयू के इन सेम के एग्जाम में बहुत सारे क्वेश्चन प्रीवियस ईयर से ही रहते हैं। तो इसलिए इस वीडियो को लाइक करें और अपने दोस्तों के साथ शेयर जरूर करें। और मैं सारे सब्जेक्ट का पीवाईक्यू सॉल्यूशन अपने चैनल पे लाने वाला हूँ, इसलिए मेरे चैनल को सब्सक्राइब कर बेल आइकन जरूर दबाएं।